Welcome to Lecture 8C, an optional lecture for this course's unit on vaccines. Quite a few of you have expressed interest in the development, regulation, and mechanism of the SARS-CoV-2 vaccines that have been approved in the past year, so I wanted to make sure that we go over this in this course. However, since this is an optional lecture, I want to stress that I will not be using material in this specific lecture as a source for final exam questions. So feel free to listen and not stress out too much about taking notes on this, and I hope that with this lecture you will learn some interesting new things about the immune response to SARS-CoV-2, the current SARS vaccines, and the regulatory processes that have allowed us to go from unknown viral outbreak to vaccine within the space of about a single year. Also keep in mind that this lecture was recorded in summer of 2021, so there may be some information that's already out of date by the time you watch this, given how quickly research in this field is moving. Let's start with a little bit of background on the clinical symptoms and correlates of severe disease following SARS-CoV-2 infection. We all know that in late 2019 there was a cluster of pneumonia cases reported in Wuhan, China, that was caused by a novel coronavirus that hadn't been reported to infect humans before. This is the beta coronavirus SARS-CoV-2, which is closely related to SARS-CoV-1, which had caused smaller outbreaks in China in the early 2000s. SARS-CoV-2 quickly spread all over the globe and was declared a pandemic in spring of 2020. As a quick bit of vocabulary, SARS-CoV-2 is the name of the actual virus that causes this infection, which we also refer to as the etiologic agent that underlies infection. COVID-19 stands for Coronavirus Disease 2019 and refers to the set of clinical disease symptoms that are observed following infection. People have been using these interchangeably and saying things that are technically incorrect, like infection with COVID-19, so just try to make sure that you keep these straight whether you're talking about either the virus or the clinical signs of disease. I'm not going to exhaustively go over the clinical symptoms associated with COVID-19, as I'm sure everyone here is pretty familiar with the main ones by now. So SARS-CoV-2 infection often leads to respiratory symptoms, such as fatigue and shortness of breath, which is observed in almost all symptomatic patients. In patients with severe disease, this escalates about a week after symptom onset, leading to something called acute respiratory distress syndrome, or ARDS, that eventually makes it so that the patients need to be admitted to the ICU for respiratory support within about 1.5 to 2 weeks following initial symptoms. There are a lot of other manifestations of COVID-19, including symptoms in the gastrointestinal or renal systems, as well as some pretty scary neurological effects in the central nervous system. Researchers are still studying how this one virus can affect so many different organs, but it's thought that because the entry receptor for SARS-CoV-2 is expressed in many different tissues, the virus just might end up setting up infection in different organs in some individuals. By now, we also know that there are a number of risk factors that correlate with the onset of severe COVID-19. Age is a major risk factor, so a lot of elderly people are at much higher risk of having severe or critical forms of disease that could kill them. Other pre-existing conditions such as heart disease, diabetes, and hypertension are also correlated with a higher risk of severe disease and death following infection. This slide highlights risk factors associated with severe disease, but I want to stress the fact that part of why this virus is so serious is because it also infects perfectly healthy younger people. Although it happens at a lower incidence, there are still patients in their 20s, 30s, and 40s that have died from COVID-19. Also keep in mind that these infections are not simply separated into binary outcomes of death versus survival, as there are a lot of people who had mild signs of disease but now have long-term respiratory or neurological impairments, and these are collectively referred to as long COVID. Since there will be millions of survivors with potentially long-term medical issues, this has the potential to be a huge burden on public health and hospital systems in upcoming years. Since this is a new virus that has never infected people before, we just don't have any data yet to tell us what the consequences are going to be in survivors, which is why it's important to not trivialize even mild forms of infection. Given all of these issues, it's critical to develop vaccines that will work globally to prevent infection. Before we get into these vaccines, let's first go over a simplified description of the life cycle or the infection cycle for SARS-CoV-2. Like most viruses, this exists as extracellular viral particles, or virions, that are made up of a spherical capsid that encases a set of nucleic acids. Because the virion does not contain all of the cellular machinery for translating proteins, the virus needs to enter host cells for replication and propagation. SARS-CoV-2 does this through expression of a structural glycoprotein on its capsid that's called spike, or S protein. Spike protein binds to an entry receptor at the plasma membrane called angiotensin converting enzyme 2, or ACE2. This interaction is required for viral entry through endocytosis and membrane fusion with the viral particle. ACE2 is expressed by cell types in many tissues, but is predominantly found on epithelial cells in the respiratory tract, which explains why this virus exhibits tropism, or preferential infection, of the human lung. SARS-CoV-2 is a single-stranded RNA virus, which means that its genome is encoded in a piece of RNA within the viral particle. Once inside the cell, the particle goes through a process called uncoding, which exposes the virus's RNA genome to the cytosol. At this point, it uses the host cell's own machinery to translate viral proteins and replicate its own genome, which are then used to synthesize newly assembled viral particles. These particles are then exocytosed into the extracellular space, and this releases many new virions and completes the cycle of viral replication. This figure from a review article summarizes the replicative cycle of SARS-CoV-2, noting the steps of the cycle that are targeted by various antiviral therapies that have been used in COVID patients. Notably, none of these work particularly well across large groups of patients with respect to increasing patient survival. So the RNA polymerase inhibitor remdesivir is often used, but its only proven effects, at least at this point, are to shorten recovery time for hospitalized COVID patients, so it may not actually improve survival outcomes. Some monoclonal antibodies against spike protein, such as a combination of bamlanivimab and etesevimab, I butchered those a little bit, these have received emergency usage authorization for treating patients since they actually can improve survival. However, the cost of treating a single patient with high doses of monoclonal antibodies is very high, so this isn't really an accessible treatment option for many patients, and this highlights the need for preventative measures such as vaccines. If you remember back to Lecture 8b, a key step in developing effective vaccines is to determine the natural immune response to infection. This helps scientists define which immune cells or molecules are associated with protective immune memory, which is something that the field refers to as correlates of protection. These are important to establish following natural infection because they can then be used to evaluate samples from vaccinated individuals to determine how well the vaccine works. Luckily, given how many immunologists are analyzing COVID patient samples at this point, we're starting to generate some promising data on the correlates of protection. If we revisit this diagram from our general vaccine lecture, there are many steps of an immune response induced by vaccines that mirror what you would expect to see during the course of natural infection. For SARS-CoV-2, we know that adequate levels of type 1 interferon is a positive prognostic indicator, which makes sense since these induce an antiviral state in infected cells. We also know that balanced levels of inflammatory cytokines and chemokines are a good thing since they help recruit and activate immune cells, but in some patients with severe pathology, they actually have increased levels of cytokines and chemokines, and this is thought to drive immunopathology in the lung through something called a cytokine storm. This is why some therapeutic treatments for COVID patients are immune modulators that can help dampen this form of uncontrolled innate immune activation. There are ongoing clinical trials to examine the efficacy of the anti-IL-6 monoclonal antibody, tocilizumab, as well as adalimumab, which is an anti-TNF-alpha therapy. People are also testing certain steroids that will suppress global inflammation, and there's some others that are being tested to see if dampening certain inflammatory signals of the innate immune response helps to control dangerous lung immunopathology. Analysis of survivors also reveals favorable data that shows that SARS-CoV-2 infection leads to expected memory responses in adaptive immune cells. This includes the formation of memory B cells and plasma cells, which generate neutralizing antibodies following infection. A lot of these antibody responses are characterized by anti-spike protein specificity, although there are also some other viral antigens that B cell memory can target. These include both IgG and IgA isotypes that can presumably mediate virus neutralization upon re-encounter in sites like the respiratory mucosal surfaces. There's also evidence for circulating memory T cells that can mediate cytotoxic responses, which may be important for eliminating any cells upon reinfection in that same patient. Although these datasets look promising so far, a major outstanding question right now is how long these forms of immune memory are maintained in people that survive infection. This pandemic just hasn't been around long enough to understand how long immune memory is maintained, and so we simply won't know the answer to this question until more time passes and we can evaluate long-term patient samples. But for now, it's very encouraging to see that the endogenous response to SARS-CoV-2 exhibits some expected immune correlates of protection. We can use these as readouts of vaccine efficacy as we start to characterize immune responses to these new vaccines. Let's next get into what these vaccines actually look like. By this point, you should understand that the viral spike protein is essential for infection since it is required for viral entry. Also, a lot of the dominant immune memory responses following infection seem to be driven by anti-spike antibodies as well as spike-specific T cells. So not surprisingly, spike has been selected as the main target for the antigen component for essentially all of the SARS-CoV-2 vaccines. Here's a structural schematic of the SARS-CoV-2 virion. Again, spike protein is located on the capsid of the virion, and the protein structure looks kind of like this rough mushroom shape, as you can see in this portion of the figure on the right. It has a receptor binding domain, or RBD, that's highlighted here in red, and this contains all of the amino acid residues that are physically involved in binding to the ACE2 entry receptor that's expressed on host cells. The current vaccine formulations are the first coronavirus vaccines to ever be developed for licensed use in humans. So coronavirus vaccines had historically been a low priority because strains that circulate in humans have typically caused mild colds, while the nastier strains of SARS-CoV-1 or MERS were relatively smaller outbreaks that could be controlled quickly. The current SARS-CoV-2 vaccine candidates come in all sorts of different forms, which fit with the main categories of vaccines that we first introduced in Lecture 8b. These include killed vaccines in Panel C, live attenuated in Panel D, and spike subunit and virus-like particles in Panels E, F, and G. There are also viral vector vaccines, which use something like an adenovirus to express nucleic acids encoding spike in host cells, and these are shown in Panels H through I. Another way to enforce expression of viral spike in host cells is through nucleic acid vaccines, which can come in either DNA forms in K or RNA forms in L. Since these are the types of vaccines that were first approved for use in the United States, we will come back to discuss them a little bit later on in this lecture. I know a lot of people are worried about how quickly these vaccines have been generated, tested, and approved, so let's talk about how this process has been done safely with vaccine candidates. We can start off by comparing how the timelines differ for traditional vaccine development versus vaccine development for the new SARS-CoV-2 products. First, traditional vaccine development is a fairly lengthy process that typically takes about 15 years. This starts with preclinical studies in mice to design vaccines and characterize immune responses to new candidates, followed by toxicology studies and production protocol development in other types of animal models. These preclinical phases usually take several years. The candidate then enters phase 1, 2, and 3 clinical trials, each of which take a few years and define vaccine safety, dosing regimens, clinical correlates of protection in humans, as well as overall efficacy. If everything in this process goes well, then you end up filing a licensing application with the FDA, and this is another process that takes a few years. And then once approved, it takes additional time to manufacture and distribute the vaccine. I want to point out that this process is extremely expensive, so at every single one of these steps, a company will also carry out economic risk assessments, just to make sure that the vaccine is working well enough to justify spending this much time and money on its development. This is a main reason why it takes so long for vaccines to be developed, since companies conduct extensive economic risk assessments upon completion of each stage before deciding to move on to the next stage. The COVID-19 pandemic has put this process into fast forward because this is an unprecedented emergency that requires rapid development of preventative therapies against infection. Now, existing preclinical vaccine research had already been established for prior SARS-CoV-1 and MERS outbreaks, which saved considerable time in the preclinical phases of vaccine development using animal models. Once the sequences for SARS-CoV-2 spike protein and protein crystal structures were published back in January of 2020, pretty much any company that had an existing platform for making vaccines started making SARS-CoV-2 candidate vaccines. This was pretty easy because if a company already had vaccine design platforms developed for other viruses, all they needed to do really was just swap in a new antigenic target for the new vaccine, which in this case was the spike protein. Keep in mind that for many of these vaccine platforms, they had either already been approved or were in intermediate stages of approval for use in humans. So it's not like all of these vaccine formulations had never been tested before. They just needed to be tested using a new antigen for a new virus. The first phase 1 clinical trials for the SARS-CoV-2 vaccines started in March of 2020. This is really incredible when you think about the timeline. So the scientific community kind of banded together to figure out the information needed to design these vaccines in a matter of just a few months. We went from not even knowing which virus caused this cluster of pneumonia cases in China to isolating and sequencing viral strains, identifying spike protein as a requirement for viral entry, identifying the receptor binding domain of spike, and then generating vaccines all within about 4 months. So this is quite impressive, and I think it's a great example of collaborative research being used for the common good. The trials for these vaccines have also been staggered, which helps speed things up further too. So rather than finishing up all of phase 1 before moving to phase 2 and then 3, each of these phases overlapped with one another to help get things moving quicker. This had been done in such a way that safety was not compromised, since these trials have enrolled tens of thousands of patients. So we have robust early-stage data sets on the safety and efficacy of these vaccines. As I'm sure you've remembered, any adverse side effects during these trials are taken very seriously, so they'll stop the trial and evaluate things before continuing, and this shows how they're still meeting rigorous FDA standards. So I understand why people may be worried about the accelerated timelines here, but these studies are still being done in very safe and responsible ways. Large-scale production for these vaccines was also started by companies even before phase 3 trials began, and this was a huge monetary risk that companies usually don't take. So remember that with traditional vaccine development, extensive economic risk assessments are conducted between each phase. However, due to the overwhelming need for vaccines, companies have moved forward through each stage without focusing on financial risk, and this has enormously sped up this process. I would say that the existing preclinical research, along with the removal of financial assessments, are primarily responsible for speeding up the development pipeline for these vaccines, and again reiterate that no corners have been cut with respect to patient safety concerns. Lastly, the licensing process has also been sped up through emergency use authorizations by the FDA. So these EUAs are based on short-term clinical trial data which have shown vaccine efficacy, and these are used to justify expedited approval of medical treatments, specifically during public health emergencies such as the current pandemic. Full clinical trials will still be completed and reviewed using all of the standard FDA protocols, but in the meantime, these vaccines have been approved since they prevent infection by a deadly virus with limited alternative treatments. Taken together, these factors all explain how we've been able to go from early preclinical research phases to approved vaccines in the remarkable timeline of less than a year. This is a lot faster than I had initially expected at the start of the pandemic, and I again want to stress that this process has still been conducted with the same rigorous safety standards as any type of clinical testing of new therapeutics. This figure is from a review article that was published in October of 2020, which means that it's already out of date. But I think that it's helpful in illustrating just how many different vaccine products there are in development for use against SARS-CoV-2. Vaccine candidates have been generated using almost every type of antigenic component that we've learned about in this course, and there are hundreds of candidates spread out between all phases of the vaccine development pipeline. It'll be really interesting to see how all of these candidates differ from one another with respect to immunological correlates of protection and how robustly they can stimulate long-term immune memory, though we're just going to need to wait a while in order to get those more long-term data sets. For now, let's look at the responses induced by the main vaccines that are approved for usage in the United States currently. As of summer 2021, there are a handful of vaccines that have received emergency use authorization in the US, although I'm sure this slide will be out of date by the time you watch it later on in the summer. This includes the Johnson & Johnson adenoviral vector vaccine, as well as the Moderna and Pizer BioTech mRNA vaccines. I've also included AstraZeneca's adenovirus vaccine on this slide since it's approved for usage in a lot of other countries. Each of these vaccines function with the goal of transferring nucleic acids encoding viral spike protein to host cells, although they accomplish this in a couple of different ways. First, the adenoviral vaccines work by using a harmless virus called an adenovirus, which is also referred to as an adenoviral vector. This virus is engineered to encode viral spike protein in its DNA genome. So when the virus is introduced into the body through vaccination, infected host cells will translate and transcribe the viral DNA, leading to spike protein production. Importantly, these adenoviral vectors are severely attenuated and cannot replicate in host cells, so they're only used to introduce target genes to host cells and are very safe. Second, the mRNA vaccines work by taking a piece of messenger RNA encoding viral spike and encasing it in a lipid shell, which is called a liposome. This is essentially an artificial phospholipid bilayer that's similar to the one that makes up the plasma membrane of host cells. Once administered into the body through vaccination, these liposomes are taken up by resident antigen-presenting cells, such as dendritic cells and macrophages, which are then responsible for translating the mRNA into spike protein. Notably, these liposomes can also drain to the lymph node where they meet up with lymph node resident APCs and kind of do the same thing. Because there are no infectious components of these mRNA vaccines, they are very safe to use. And as a side note, mRNA and liposomes are relatively unstable molecules, which is why these vaccines need to be kept in very cold freezers for storage. It's worth noting that although both of these methods use nucleic acids, there's virtually zero risk of these ever incorporating into a host genome. Adenoviral DNA and liposomal mRNA are just short segments of nucleic acids that are taken up by target cells and degraded shortly afterwards, and it's very highly unlikely that any recombination event would ever occur with host genomic DNA. I know that this has been brought up as a so-called safety concern, at least in the press recently, but I want to stress just how difficult it is to successfully target genomic recombination events, even if that's what you're specifically trying to do in the first place, because this is the entire field of genome engineering. So this is a highly unlikely risk of either of these vaccines. Whether through adenoviral delivery of DNA or liposome-mediated uptake of RNA, the end result is that cells in the vaccinated tissue synthesize viral spike proteins that are then displayed on the cell surface. These can present antigen to activate spike-specific T cells, as well as make spike available for binding to B cells. Therefore, these vaccines can induce both T and B cell mediated immune responses, leading to the generation of protective immune memory. For each of the mRNA-based vaccines, I wanted to go over the clinical trial data that justified the emergency use authorizations for the Moderna and Pizer BioTech vaccines, because these are just quite obviously beneficial results. So both of these figures are taken from phase 3 clinical trial results for Moderna on the left and Pizer BioNTech on the right. Each of these studies involved tens of thousands of trial participants who were randomly split into two groups, one that received placebo injections of liposomes with nonsense RNA that didn't encode any sort of protein, and then another group that received injections of liposomes with RNA encoding the spike protein as a target antigen. Trial participants were then followed out for a few months to see which participants got infected with SARS-CoV-2 as confirmed through qPCR testing for viral RNA. Both of these graphs plot the cumulative incidence or event rate of confirmed infections on the y-axis versus time on the x-axis, and that's time since they got their first shot. For both vaccine candidates, you can see that the group of participants that received the actual vaccine pretty much flatlined with respect to infection, as these curves stay flat with an incidence rate close to zero, while the placebo groups gradually rise upwards as members in that group got infection through community transmission of SARS-CoV-2. These studies do assume that both groups of participants were equivalently exposed to the virus, which of course is impossible to validate. And this is why these have to be such large trials, including tens of thousands of patients, to try to account for that. These flatlined curves are pretty much the best case scenario that you could hope for when testing the efficacy of a vaccine, and are how we calculate the efficacy of these vaccines to protect over 90% of immunized individuals. There are obviously outstanding questions from these trials related to how long vaccine-induced protection lasts, and how well immune correlates of protection line up with those seen after natural infection. But from this strong phase 3 data, the FDA approved both companies' requests for emergency use authorizations. It's a hopeful start to see that these fast-tracked vaccines appear to be working exactly the way that we hoped that they would, based again on what we know about the human immune response to immunization. I want to end today's talk with an overview of some of the next steps for the SARS-CoV-2 vaccines. Having these first few vaccines approved for emergency usage in adults is a big step towards getting society moving back towards some semblance of normalcy after a very difficult past year. But there's still a lot of work to do on the vaccine front. First off, even for vaccines that have received FDA emergency use authorization, companies are still working on optimizing vaccine formulas to do things like decrease side effects, improve their stability, and reduce dosing regimens to only need one dose instead of two. Vaccine optimization also includes plugging in new spike or other antigen sequences to account for new viral variants that have emerged since the original vaccines were developed, or to identify antigenic epitopes that stimulate robust neutralizing antibody production that works across all variants or all strains of the coronavirus. There's also the question of ramping up manufacturing and distribution to immunize people around the globe. And this is a huge problem right now, since most of these vaccine products are only available in the richest countries that are best equipped to handle the pandemic, at least infrastructure-wise. And so we're worried about many countries not having equitable access to these vaccines. The vaccine pipeline is going to be a process that goes through multiple iterations of optimization and improvement, and there are going to be a lot of different vaccines on the market. Although these may vary with respect to things like their dosing regimen and storage conditions, every vaccine that receives emergency authorization will function similarly, at least with respect to the only readout that matters at this point in the pandemic. And this is that they reduce incidence of SARS-CoV-2 infection, and that they accomplish this safely with minimal adverse side effects. And again, these are important safety concerns that are also being closely monitored. It's important that we move forward quickly with mass vaccination efforts, because the longer we allow viral transmission to occur within a population, the greater the likelihood we have of selecting for viral variants that escape antigen recognition by vaccine-induced immunity. And so these would be variants that essentially evade vaccination responses. So far, all of the vaccine data has shown that neutralizing antibodies are still effective against the more transmissible variants that have emerged across the globe, and this is reassuring. But the concern is that as transmission continues in unvaccinated populations, we are essentially giving the virus more opportunities to replicate and acquire mutations that could eventually allow it to escape pre-existing immune memory responses. Considering all of the information that you've learned today and in our previous lecture 8b, I hope that you've learned that the SARS-CoV-2 vaccines confer host protection from infection, and that they do this safely even though vaccine development was fast-tracked over the course of the past year. This is an incredible achievement that has required collaboration between basic research labs, clinical research teams, and pharmaceutical companies. And I really want to emphasize how the scientific community has banded together in response to the pandemic to try and get vaccines out to the public in such a short time frame. For future steps, we can expect to see multiple iterations of vaccine formulas that optimize their ability to confer long-term protection while minimizing side effects. There's also a pressing need to ramp up production and distribution enough to make the vaccine accessible for everyone, which is an important consideration for healthcare equity both within the United States as well as across the globe. Lastly, we need to speed up vaccine rollout to try and immunize as much of the population as possible in order to decrease the likelihood of selecting for more transmissible or virulent strains of the virus, because this is kind of exactly what we're doing as states lift restrictions and you get unvaccinated people continuing to do things like gather in social groups without distancing or wearing masks. That's it for vaccine lectures in applied immunology. I hope that with the information we've covered this week, that you can take what you've learned of the immune system in order to fully understand how vaccines work, and that this information helps you address any safety concerns regarding both the SARS-CoV-2 vaccines as well as other vaccines. Lastly, here's just a quick reminder to check Canvas for reading and discussion assignments this week, as well as the extra credit quiz for week 8. Good luck with your work this week, and see you next week for our week 9 module.